This is Lynn from my research I did on cloning endangered gray wolves from somatic cells collected post-mortem. The gray wolf originated in Korea has been declining in population since the 1960s. To keep them from going extinct, we have been drawn to the idea of cloning the gray wolf in hope to gain a better understanding of somatic cell nuclear transfer. Reproductive technologies like embryo cryopreservation (AL) and somatic cells nuclear transfer (SCNT) have been used as a potential preservation of endangered animals. SCNT, also known as cloning, is a technique where the nucleus of somatic cell is transferred into another cell without a nucleus. This could potentially help restore endangered and extinct species. In order for cloning to be successful, oocyte cytoplasm need to support the development to the blastocyst stage of embryo. Some successful blastocyst development using interspecies SCNT has been reported in species like argali, giant panda, water buffalo, and bongo. In addition, the live offspring production by interspecies SCNT has been reported in Gaul, Morphlin, and female gray wolf. The primary objective of the study was to demonstrate the use of ISCNT using somatic cells collected post-mortem from an adult gray wolf. Furthermore, we researched on whether the mitochondrial DNA, mtDNA, of the clone wolf matches the oocyte donor dog or the nuclear donor wolf. Second, the amount of telomeric DNA, also known as ATD, to receive from the nuclear donor wolf. We used a mixed breed of female dogs between the age of 1 through 5 as oocyte donors and embryo transfer recipients. The donor somatic cells were obtained from the abdominal skin fibroblast from the 3-year-old male gray wolf 6 hours after death. The oocytes were obtained from oviducts around 72 hours after ovulations, and the single cell was placed into an enucleated oocyte. Pregnancy was monitored ultrasonographically every 2 weeks after confirmation. As a result, out of 17 recipients' dogs, only 4 recipients were pregnant 23 to 25 days after embryo transferred, with 6 pups delivered. That put us at pregnancy rates of 23.5%. 2 dead pups were prematurely and spontaneously delivered by one of the 4 recipients on day 58. One of the dead pups had no pathological lesions, but the other has anoestresia and tail agenesis, with severe liver and moderate kidney congestion. The rest of the pregnant recipients were delivered by C-section on day 60, with 4 clone wolf pups weighted 450, 190, 300, and 490 grams. The 190 grams puppy died within 12 hours after it was born. It was disclosed that it had a defect in the corpus callosum and the hippocampal commissure of the brain. The 3 male clone wolf pups are still alive, but one has female external genitalia, no testes, and a penis. The 6 clone male wolves and the nuclear donor wolf had identical microsatellite patterns for all loci, and the female recipients were not similar. Furthermore, the 6 clone wolves' mtDNA sequences were identical to the domestic dog oocyte donor. The 3 clone wolves that are still alive had the same number of 78 normal diploid chromosomes and a pair of sex chromosomes as the nuclear donor wolf. Comparing the ATD of the 3 clone wolves with their nuclear donor, they had a similar or lower ATD. The interface of ATD of clone C was 1.93, similar to the nuclear donor at 1.91%. But the clone D and the clone F were different. The clone D had 1.14% and clone F had 1.46%. Our percentage of the pregnancy rate in this study is higher than the previously recorded at 16.7%. We knew more accurately the time of ovulation, leading to more successful results. We collected around 70% mature oocytes compared to 50% from a previous experiment. We had greater accuracy, minimizing our time handling the oocyte in the vitro, and we also calculated the number of embryos needed to create pregnancy, allowing us to place at least 13 zygotes for each test. During this study, 4 recipients had detected heartbeats, but one gave birth to 2 dead pups, as well another clone birth at low weight and died relatively quick. It appears as though there are many defects of cloned mammals through culture cells. The defects can be caused by nuclear transfer technique, in vitro manipulation, nuclear reprogramming, culture donor cells, or quality of the oocyte donors. Further testing must be done to further understand why these problems occurred. When cloning the male gray wolf, we had hybrid embryos due to the lack of gray wolf oocytes. Thus, we placed nucleus DNA of the gray wolf into a female domestic dogs, providing the empty DNA. The D loop has to be identical to the donor for the clone to be genetically similar. There are many controversies around cloning using a surrogate because there are many failures that are unknown. For example, restoration of cellular structure does not always get completed by nuclear donor cell. The ATD in the 3 live pups derived from the skin fibroblasts were less than the nuclear donor wolf. The length of telomere in cloned animals could be due to species differences and/or differences in nuclear transfer techniques or donor cell types. We could not compare the ATG in the clone wolf pups with the control animal because no animals were available due to endangerment. The wolf pups appeared to be healthy despite the shortage of telomere length. However, further research on whether telomere length has an effect on the lifespan of the animals is required. To wrap it up, we believe that our experiments in the cloning of the gray wolf using in vivo mature oocytes collected from domestic dogs were successful. We showed that nuclear donor cells from the postmortem wolf could be used genetically cloned by interspecies SCNT. One critique I have is we should take consideration of the maturity of the donor oocytes and tissue's origins, because these can affect the pregnancy rates. We should also try to improve our technical skills so that we can get a higher percentage rate of mature oocytes and quality of the zygotes. There are many options for our next research experiments. One of them should focus on how to increase the pregnancy rates and to study the causes of abnormalities in the wolf pups. We also know that the lifespan of the clone animals is short. Thus, we should do more research on whether the telomere length alteration through SCNT have any effect on the lifespan.